Good morning, everyone. On behalf of the organization committee, I would like to thank you and welcome you in our webinar entitled COVID-19 Impact on the Lebanese Setting. We are very happy today to successfully join 425 registered participants from Lebanon and from abroad. This is in addition to 17 national and international speakers from the Arab region and from Lebanon. Without any further ado, I'm very honored to welcome our Dean, Professor Raja Fakhouri, who will be giving us the welcome speech. Dr. Fakhouri, the screen is yours. Thank you.
Thank you, Dr. Saeed. Good morning. It gives me an immense pleasure and great honor to welcome you all to this virtual symposium hosted by Beirut Arab University Faculty of Health Sciences, with the participation of many colleagues, collaborators, and partners from different universities and hospitals in Lebanon, Qatar, UAE, KSA, and France. Thank you all for joining us today. This scientific meeting is a great opportunity for all of us to exchange experience and knowledge, and to establish research collaboration with the clear objective of advancing research synergies in the fight against COVID-19 that changed the globe. A year ago, the WHO declared COVID-19 as a global pandemic. During the last year, the life of almost every person on this planet has been greatly affected by this virus, whether it's social, economical, or health. We have been all surrounded by a mountain of uncertainties. In his last WHO press conference, Dr. Tedros, the General Director of WHO, declared that some countries in Europe, Africa, and USA are seeing spikes in cases with multiple factors driving the transmission risks. Today, we have more than 2 million deaths worldwide and a new variant appearing in multiple countries. Dr. Tedros added, health workers are exhausted and health systems are collapsing. We are all aware that there is an immense pressure on hospitals and health workers in our beloved country. Therefore, there is an urgent need for a national committee to call for an urgent emergency situation, because the more the virus is suppressed, the less opportunity it has to mutate. We all need to share our expertise and resources to help this virus, to stop this virus from killing our family and friends until we reach the end of the tunnel. Please allow me on this occasion to thank our physicians, our nurses, and all health workers for the great effort and sacrifice amidst those difficult times. Thank you, colleagues, Dr. Saeed, Dr. Hiba, and Dr. Ahmed for organizing this event. Wish you all a very successful and enriching webinar. Thank you.
Thank you, Dr. Raja. Thank you very much, and welcome everybody to our today's webinar. First thing I would like to add is the following. For all the audience, please stay with us until 13:35 today. This will be around, it will be 6 sessions. At the end of every session, there will be a question and answer. During the running of the presentations, all the audience can ask any question at any time. We will try to answer all as much as possible during the question and answer session. After the last session, we'll have also a roundtable. We will be talking about everything to do with what's going on with the updates concerning the corona vaccine, and we will have an open discussion also about that. From now, after a few minutes, we will start also a poll. We'll ask you a question, actually 2 polls, 2 questions, and about the vaccine, about other things. And in the roundtable at the end, we will open the answers and we will discuss your views in this concern. So please try to do your questions to the presenter during their time and during that session, so that we can do a question and answer, so that we don't go back to the previous sessions. Thank you very much. We will start with the first session 1 with the moderator, Dr. Ayman Al-Khatib. The screen is yours, Dr. Ayman.
Good morning, everyone. Thank you all for attending, and hope you are all safe and sound. Actually, I'm honored to present the keynote speaker of this webinar today, Professor Ali Hajir. Professor Hajir did his BS in Medical Sciences from Yarmouk University in 1985 and his master's degree in immunology from the same university in 1988. In 1991, he obtained his PhD in immunology from the University of Manchester, where he continued to fellowship in histocompatibility and immunogenetics. In the year 2000, he obtained FRC Path in H&I, London, and in 2010, he obtained the American Board of Histocompatibility and Immunogenetics, the ABHI. In 2001, he joined the Ministry of National Guard Health Affairs as Director of the Immunology Immunopathology Lab. In addition, he is currently the Assistant Dean, College of Medicine, KSAU-HS. Professor Hajir published more than 200 peer-reviewed publications. Professor Hajir, the webinar now is yours. The screen is yours.
Good morning, everyone. Can you hear me?
Yes.
Yes, we can.
It gives me great pleasure to join you this morning in this fascinating research day. I would like to thank the organizers, all my friends there, Ahmed and Saeed, everyone there. Thank you for the invitation. Thank you, Professor Raja, for the invitation. And really, this is a great honor to join you this morning. In my talk today, I will try to take you through the up-to-date knowledge on antibody responses to SARS-CoV and touch on the diagnosis and slightly on the vaccine at the end. So, as we know, coronaviruses, they belong to an RNA, single-stranded RNA viruses. They are enveloped. The new virus, SARS-CoV-2, belongs to the beta coronaviruses. With it, we have also the MERS-CoV, which is the Middle Eastern Respiratory Syndrome virus that was found in or discovered in Saudi Arabia, and the original SARS that was described in 2003. The main host of these viruses are the bat, with intermediary host for the SARS-CoV-1, the wild cats, and for MERS-CoV, we have the camel. And it is speculated that for SARS-CoV-2, it is the anteater, the pangolin, being the intermediary host for this new SARS-CoV virus. The genomic organization is very similar between the different coronaviruses. They have the S antigen, which is the spike antigen, which binds to the receptor on the cell and gets inside the cell to be invading the cell and get replicated. We have the M antigen, the E antigen, the N antigen. So these are the envelope, the membrane protein, and the nucleocapsid protein. These are the major proteins forming the virus. Now, is there a cross-reactivity between SARS-CoV-2 and the other beta coronaviruses? There is a strong cross-reactivity with SARS-CoV-1, which was completely disappeared from our world. And there's about 90% homology between SARS-CoV-1 and SARS-CoV-2 at the N antigen and S2 antigen, about 52% with the S1 antigen. However, with other beta coronaviruses, the cross-reactivity is very little, so 30 to 40%, and even lower with the alpha coronaviruses. And this can be demonstrated in this graph where people looked at serum from different coronaviruses. You know, the common cold that we experience in our normal life. We have the common cold coronaviruses, the human coronavirus infection, MERS-CoV, SARS-CoV, and you can see that only SARS-CoV-1 can be some sort of cross-reactivity with SARS-CoV-2 virus. This slide was updated yesterday, We know SARS-CoV-2 infection was identified in China in 2019. It spread to most countries nowadays. As of yesterday, there are more than 100 million infected people. So far, we have about 2.1 million deaths with a fatality rate of about 2%. What about antibody responses to this virus? To understand this figure that I'm showing here will help us in understanding the diagnosis of this infection. So as with many acute respiratory infections, we start to have the RNA coming up, detected in the respiratory samples, and it takes some time for the antibodies to appear. So in the first 1 to 2 weeks, it is difficult to use antibodies in the diagnosis. And the only way to diagnose acute respiratory infections is to use the RNA-based assays. However, after 10 to 15 days, we start to see the IgM, the IgG, the IgA antibodies. And with time, the RNA levels start to decline. Interesting enough, there is a major phenomenon with coronavirus infections, and I will show you a big study about that in the common cold coronaviruses in a minute. But with SARS-CoV-2 and with MERS as well, it is interesting to note that with time, the antibodies start to disappear. And you see that with asymptomatic, it even disappears much faster than the symptomatic cases. And you will see higher levels of antibodies with the symptomatic than the asymptomatic. In our hands, we have seen the antibody levels disappear within 40 days of the end of the symptoms in many people. And this slide just described that. So in the asymptomatic, about 60% of them, they will lose the antibodies. And in the symptomatic, I'm sorry, in the asymptomatic, about 60% of them, they will have the antibodies, which will disappear. And in the symptomatic, 87% of them, they will have antibodies. Can you detect antibodies in everyone? Unfortunately, antibodies do not sort of show in every individuals. So 40% of asymptomatic individuals become seronegative, and only 12.9% of the symptomatic become negative in about 8 weeks post-infection. There was a very nice paper in New England Journal of Medicine describing the rapid decay of the SARS-CoV antibody, IgG antibodies with time. And almost within 3 to 4 months, most patients, they seem to lose their antibodies. So when do the antibody to SARS-CoV appear and disappear? In most cases, the antibodies, they appear about 10 to 15 days, and they start to disappear within about 40 days. And this is very interesting. And that applies to IgM, IgA, and IgG. So is there a relationship between the antibody level and disease severity? I would like you to focus on this slide, and this is a very interesting slide. And unfortunately, many people do not really sort of focus on this issue. The first thing is that in the severe cases, we have more antibodies than the mild cases. And this is for IgA, anti-RBD antibody. We have the IgG in the same way, where severe cases, they have much more antibody levels than the mild cases. This is the first phenomenon. The second phenomenon is that, very interestingly, the IgA antibody appear to have higher levels of the antibodies compared to the IgG. So the titers SARS-CoV patients develop with IgA are usually much higher than IgG. And just I would like to stop here for a second and remind you that SARS-CoV is a respiratory disease, and the main antibody that fights respiratory infection is IgA. Unfortunately, many people they focus on IgG and IgM. Even in the convalescent plasma for treating patients, they use IgG, IgM antibody levels. They don't look at IgA. And from this stage, I would like to really emphasize that IgA is a better marker for the disease, and I believe that it is the antibody of choice for treatment. And in this slide, it echoes the same sort of message. In the mild cases, the IgA are lower, while in the severe cases, the IgA titers are much, much higher. And when it comes to IgG, the titers in the mild cases are lower, while in the severe cases, you will see much higher antibody titers. And still, IgA have at least 2 to 3 times the levels of IgA when it comes to titers. This is a very fascinating slide and very fascinating study where the authors looked at the neutralizing antibodies and they wanted to see, can the antibody titer predict outcome? I'm having something coming on my screen. I don't know how to get rid of it. Yes. I'm sorry about this. So in this first part of the graph, you have IgG anti-RBD antibodies, and those titers, they predicted with big accuracy the death. So death being as the outcome, the higher the titers of the IgG, the higher the risk of death or intubation, followed by hospitalization. And when it comes to IgM, in a similar fashion, but the titers for IgG are much higher than that for IgM. And IgA also predicted death, intubation, and hospitalization. This is a very interesting phenomenon. When you have patients hospitalized, following up antibody levels will help you in predicting outcome on those patients. Which antibody assay shall I use? There are several assays in the market. Unfortunately, we have seen new companies coming to the market with no experience in the past in producing kits. And I would like to make a warning here that do not believe in any company that produces a kit for the first time and comes to you and trust the result. I'm a believer in experience. If I want to buy something, I'll buy it from a reputable company, someone that have been producing kits for some time, not just a company coming from nowhere and giving you a kit for the diagnosis. In this paper, people looked at full automation versus manual, and they found that S antigen and N antigen based assays, they are better than other assays, and they found that high throughput automation are much better than manual assays. So fully automated immunoassays have higher sensitivity than the manual assays. Those usually use S and N antigen. I will come to that when I talk about the vaccine. And they are better sensitivity and specificity than the manual assays. So which antibodies are important in COVID infection? As I said, IgA is the major player here. Unfortunately, many people, when it comes to SARS-CoV, they use IgG and IgM only. I strongly suggest using IgA in the algorithm for follow-up, and even if you want to use it in diagnosis and predicting outcome. The slides demonstrate again that IgA levels, they persist higher than IgM and they stay longer than IgM. So if you pass the RNA detection and you have a negative RNA and you highly suspect a patient to have the disease, I would recommend using IgA antibody assays. What about rapid assays? As I said, many people, they are now in the market producing different kits for the detection of antibodies and even antigens. And they have never ever in the past produced a kit. And in any case, results shown that rapid assays are of very low sensitivity, 10 to 55%. And usually these kits, they will be positive when you have very high antibody titer. So if you are to detect early antibody, rapid assays cannot do that usually. And studies have shown that their sensitivity starts to increase after the 11th day of infection. And this is justifiable where you have higher titers of the antibody. Are the antibodies to coronavirus protective? This is a study that I encourage every one of you interested in coronavirus infection to read. This is a recent study, appeared a couple of months ago, where they have looked at seasonal coronavirus, the common cold, and they investigated for 35 years. Can you hear me? 35 years. They investigated the antibody responses to the seasonal coronavirus, the coronavirus that we experience every day in our own life. Look at this. They found that patients, after 35 years, patients in every single year, they experience 2 to 3 to 4 to 5 episodes of infection. Not only that, look at this fascinating slide. This is a very, very fascinating slide where they have looked at antibodies to measles over the 35 years. These dots, they represent individuals followed in this study, and the same individuals, they looked at their measles antibody, and you can see that how tight the titers are. They don't change that much. And when it comes to the other types of coronaviruses, as you know, seasonal coronavirus is not just one type. There are 4 or 5 types of seasonal coronaviruses. And their level of antibodies over the 35 years, they keep going up and down, up and down, up and down. And that tells you about 2 things. One, that the antibodies to corona are not stable. 2, the antibodies to corona are not protective. So are they protective? There is a phenomenon that was described recently, and it's in the SARS-CoV-2, and they found that antibody-dependent enhancement of infection with SARS-CoV-2. I will not dwell on this, but the idea is that when the antibody binds to the virus, it helps it to get inside the cell. So it facilitates infection. And this is usually, I believe, this is usually with the low avidity sort of antibody titers and low titers antibodies that will happen there. So what does this have on herd immunity? When we talk about rapid decay of the antibody virus, what about herd immunity? We heard about the herd immunity. And herd immunity, to achieve it, you need majority of the population to be protective, have the immunity, in order to stop the pandemic. And they calculate for the herd immunity to happen, it's 1 minus 1 divided by R naught. And in this case, they predicted that about 50 to 60%, you have to have 50 to 60%. I believe it is more than that to achieve herd immunity. And this is the idea behind the vaccination program that people are using all over the world. How can we utilize serology in SARS-CoV infection? As I said, this is acute respiratory infection, and you cannot use antibodies to detect acute infection at its early stages where the patient is highly infectious. You need to use RNA, but you can use it to follow up on patients. You can use it to when you give convalescent plasma, understanding the immune response, epidemiological studies, in diagnostics. You can use it, especially when it comes to RNA negative. So the patient passed the acute phase, and the RNA cannot be detected, and you're still highly susceptible. You can use it. You can use it to predict outcome, and definitely antibody levels predict outcome in hospitalized patients. Finally, we hear now about the vaccine, and we know that there is Pfizer and Moderna. These are the top sort of utilized vaccines so far. and both of them are mRNA based, both of them are S antigen based. And I want you to take the analogy of hepatitis B infection. In hepatitis B infection, we use the vaccine based on the surface antigen. So if you want to know a patient or a person who was vaccinated with hepatitis B S antigen, whether they are immune or not, you do S antibody for the hepatitis B, and the same here. So if you want to know whether a patient vaccinated has the immunity or not, or whether the patient has immunity due to infection or due to a vaccine, you do N antigen based assay versus S antigen based assay. In a vaccinated patient, you'll have S antigen positive, while the N antigen will be negative. In a previously infected patient, you'll have the S antigen and the N antigen being positive. I think this is my last slide. In conclusion, natural infection with SARS-CoV may not give rise to long-term immunity. And this has big implications for the vaccine. Can I take the vaccine once and that's it? It will give me forever immunity? I doubt that. I believe the vaccine will have the same story as the flu vaccine, where you may have to take it every 6 to 12 months. This is a personal judgment, not based on data, because no data is yet available for the vaccine people, vaccinated people. Serum IgA antibody is very helpful in epidemiology and in diagnosis, and even in convalescent plasma, I would predict. Antibody titer are linked to outcome, and they must be used for that. And antibodies detection can support a PCR test when it comes to post-acute phase infection. I think this is my last slide. I would like to thank you for listening, and I'm happy to take questions because I have to go to the hospital. Thank you.
Thank you, Doctor. It was a pleasure having you.
Thank you, sir. Thank you so much. I hope there was something new in my talk today.
Definitely.
Thank you so much, Professor Hajir, for this amazing presentation. We are waiting for some questions.
Dr. Hajir, thank you for the wonderful presentation and for the, most importantly, for the scientific importance. I would like to grab the opportunity to ask you, do you believe in the conspiracy theory about vaccinations? Do you think that some people from third world country will be the mouse lab for trying, for seeing if the virus might integrate the genome DNA?
I must say I'm not a believer in the conspiracy theory, and to start with the virus itself, I believe that these are natural infections, and we have these pandemics happening. In the year 1918, we had the Spanish flu, and that stayed with the humanity for about 3 years. God knows how long this will stay with us. Based on the genomic structure of the coronavirus, I doubt that this is some manipulated type of virus. As for the vaccine, I don't think there's any conspiracy behind it. People are doing their best to find a solution. And I must say that we have a problem with the coronavirus where we have a conflict between the three entities: economicians, politicians, and scientists. The politicians, they look for a renewal of their terms. The economicians, they don't want to have a problem with the economy, and they want people to get sort of employment, etc. And the scientists, they usually do not believe in the politics and the economics, and they want to find a scientific solution. So is the vaccine a conspiracy theory? I believe people, they tested it on themselves before they sent it to us. Shall I take the vaccine? I encourage everyone to take the vaccine. Will this nanoparticle have any implications in the future? I tell you, this is the first time it was used. if you are having doubts about it, use another type of vaccine, like the attenuated whole virus, for example. There is AstraZeneca vaccine that will come up soon. It's out there, but it's waiting federal approvals in the UK and the US, and that is based on a virus particle containing this antigen. I cannot remember now the Russian and the Chinese, but I believe some of them, they have the attenuated viruses. So if you are not, if you do not believe in the nanoparticles, or you think that it might have something in the future, I advise you to take attenuated vaccine, for example. The issue is people are not coping. Even the States, even America, even the England and Europe, they are not coping. And even in our countries, I believe Lebanon is really suffering from a big hit with the virus, and people are losing their loved ones. And we have to do something to stop it. I was interested in the announcement made by Tony Fauci yesterday, where he said people must use double face masks, and he believes that this will protect us more than anything else, at least for the time being, until the vaccine is being available for everyone.
Doctor, thank you very much. It was really a pleasure to have you. I have a really question. I'm just going to steal this question for a moment. Doctor, really, when do you think will be the time when the mutation is changed enough so those vaccines we have created, they're not useful anymore? Especially the attenuated, you know, the regular ones, the standard ones we used to have, and it takes months and months to generate. I know the AstraZeneca, I know the Pfizer, they are preparing themselves for something like that, but do you think we will have that time coming where the vaccine, the virus have changed enough so the vaccine is not working anymore?
The mutation, I did not touch on this, but hopefully some of other colleagues will touch on this later on. Coronavirus is RNA virus, and RNA viruses are known to have a very inaccurate replication. They have low proofreading polymerase. And that characteristics, unlike a human, for example, or other organisms, where the polymerase is a proofreader. So when it copies, it makes sure that there is no mutation. In RNA viruses, like flu, for example, like HIV, like corona, the virus keeps producing new mutations due to non-proofreading sort of issue with the RNA polymerase of this virus. So the virus will keep producing new mutations. Now, single point mutation, will it require a completely new vaccine? I doubt that. I believe that the current vaccine, will still produce immunity that can neutralize the antigen, because the single mutation will have an implication in terms of binding the virus to the cell. But the immunity, when you produce antibody to this antigen, it will produce antibody to different epitopes. Maybe it will miss that epitope where the mutation is, but it will not miss the other epitopes. So I believe it will still work. طبعا companies, when it comes to pharmaceuticals, they are not angels. They want to make money, and this is their opportunity now. The opportunity now is to make money, and they will start producing version 1, version 2, version 3, and that is nature of life. But shall we panic? I think we have enough problems to panic for, and I would say not to panic, and I would say I believe the vaccine will work, and we should really take all the measures. If we cannot have the vaccine, we should encourage everyone in our families and our friends, our colleagues, to keep the mask on at work, indoor, outdoor. It's very important to protect the community.
Thank you so much, Professor Hajir. I would like to ask you if is it possible for you to stay with us till the end of the session, because we have a lot of questions.
طيب أنا I have to go to work, لأن أنا بعدني ما رحت عندي مرضى, I have to go to work. لكن I'm happy to join you and from my mobile phone, and I'm happy to answer a question. What time do you expect the question time at the end of the session?
We still have, I guess, next.
No, just 10 minutes, Doctor.
Yes, 10 to 15 minutes, I guess, right, Dr. Ahmad?
Yes.
Yes.
Yeah, I'm happy to stay 10 to 15 minutes.
Thank you.
Okay. Thank you so much. I would like just to mention that we are now like 150 attendees and panelists in the webinar, and now I will present our next speaker in this session, Professor PATHOGENOMICS-STokajian. She is a professor of microbial genomics. She joined LAU in 2005 as a full-time assistant professor of microbial genomics, and in 2019, she was promoted to the rank of full professor. Her research interests lie in using next-generation sequencing for the molecular characterization of pathogenic bacteria, studying the molecular epidemiology of outbreaks, and implementing, developing, and introducing emerging computational biology and bioinformatics tools for the analysis of genomic data. Research in her lab involves an interdisciplinary approach using microbial genomics, cellular microbiology, and bioinformatics to understand microbial pathogenesis. She is currently targeting the in-depth genomic analysis of antibiotic-resistant bacteria, which are acronymically labeled as the ESKAPE pathogens. Additionally, and with the surge of the COVID-19 pandemic, her lab using sequenced genomes was able to identify functional domains in SARS-CoV-2 and is currently looking into the phylogenetic networks of SARS-CoV-2 genomes sampled from across the country. Professor Tokajian co-authored over 80 peer-reviewed articles, served as a reviewer for several publishers and grant agencies, and is serving as an editor on several top-tier journals. Professor Tokajian, please, you can proceed now.
Thank you very much. Thank you for the nice introduction, and thank you for all the organizers for putting this session together, which I think is very important during this difficult period of time where we are suffering from the pandemic and the consequences of the pandemic. So today I'm going to try to summarize as much as possible, as the time allocated is really not enough to cover the ongoing project in my lab, but I'm going to do my best to summarize the most important findings that we so far have when it comes to the molecular characteristics and the molecular epidemiology of this important pandemic. So first of all, let me just briefly and in a very fast way introduce you to what we do and our lab. So we are a pathogenomics lab, as I said, and as kindly was introduced, we do a lot of genome sequencing. We use different platforms. We are the only one in the region using the 4th generation sequencing platform, which is currently being used extensively throughout the world. The nanopore sequencing with the pandemic, especially, I'm sure you are aware that England is really the pioneers nowadays in tracking the evolution of the virus, and this technique and this approach was developed by the scientists, and there is now a big consortium with which we are collaborating in order also to help us in tracking the evolution of the SARS-CoV-2 as it is spreading in the community in Lebanon. So this is really an interesting time for people in infectious diseases and in people working in molecular epidemiology. Nowadays, you know that, and after, of course, going through a number of outbreaks throughout the 21st century, starting with SARS-1, and then we went to MERS, and now we're coming to COVID-19, and we know that that we had the virus appearing early last year, and then eventually the WHO declared a health emergency status, and this is where things started to really accelerate when it comes to the spread of this virus. Sequencing the virus is very important. This is part of tracking the changes that are being introduced. This actually would also help us to understand whether nowadays the vaccines that we have would still remain, if you want, functional or not. As you know, a month ago, we identified, in collaboration with the Ministry of Health, we received samples that were collected from travelers in December, late in December, and we were asked to sequence to check whether the new variant was introduced in Lebanon or not. And the sequencing outcome was yes, the UK variant was actually carried by one of the passengers. So this is very important, and I cannot stress enough, and this is what's happening worldwide now, the need to at least keep on sequencing 10% of the viruses within the community. This is the only way that you can detect the variants, and this is the only way that you can understand the impact of treatment, especially if you are using monoclonal antibodies. You need to be aware of the types of strains and variants that you have, and of course, to keep up with the changes in order also to find ways to tweak the vaccines that are available. Without these two important approaches, we cannot really reach to a point where we are faster than the virus. Currently, the virus is faster than us, but if we do what others are doing, sequence, look at the changes, change the monoclonal antibodies that we are using or relying on, and combine that with changes in the vaccine, this is the only way to get out of this pandemic. So at the beginning of the outbreak, of course, because we are a computational lab, and back in February, when we only had 500 sequenced genomes, we decided to start looking at those genomes and try to find the properties at the molecular level and try to model and in a way understand the pathogenesis of this new introduced, if you want, virus into the community at large. So what we did was, first of all, we uploaded or downloaded all the available genomes, and we started looking at the different parts of the virus genome. And I'm sure you are aware that this is a virus that has a genome that is RNA, of course, 30,000 base pair. Compared to other RNA viruses, this is bigger than the usual RNA viruses. And yes, it does evolve. However, the rate of and how it's evolving is not as fast as other RNA viruses, because although, yes, RNA viruses introduce a lot of mistakes as they are replicating their genomic material, but this virus in particular has more proofreading activity compared to other viruses. However, you might say, what's happening nowadays with all of these variants? It's normal. We started to understand the virus. We started to use convalescent plasma, monoclonal antibodies. There are different treatment approaches now that are putting pressure, selective pressure on the virus, and hence we do expect that such variants will continue to roll into the community as we use more and more approaches to try to fight it. This is very normal, and this is what happens in every pathogen. So at the time we looked at the genome, there were so many studies being done, especially ones that are targeting the spike, but we were actually interested in the ORF3A. Why? Because there is a lot of virulence and pathogenesis that is linked to this particular part of the genome of the virus, which is really not that big, but yet it is really interesting with its different domains. So we decided to upload those genomes, align them, compare them with the genomes of the virus that are linked to the bat, to the pangolin, to SARS-CoV-1, and see if we can deduce anything when it comes to the importance when it comes to virulence and pathogenesis. By the way, this part, the ORF3, it has the ORF3A, but it also has the 3C and 3B, and recently, a few weeks ago, they found a novel overlapping gene, which they referred to as 3D, and now people are studying its importance. So there are a lot of regions that are considered to be overlapping genes, and one of them is the ORF3, within which we have overlaps. We knew that we have overlaps with the 3C and B, but now We also have one from 3D. And what's interesting about it is that if you compare this new overlapping gene with that in pangolin, you will find that the only two actually viruses having those overlapping genes is the SARS-CoV-2 and the pangolin, which, you know, the hypothesis says that this was really one of the intermediary stages in the development of this virus until it became capable of jumping the species barrier and infecting humans. So our study, the main aim was to look at the ORF3A, compare it to what was known previously with SARS-CoV-1, try to understand how it is linked to the virulence and pathogenesis of this virus, and to look into mutations, especially, of course, the non-synonymous mutations, in order also to better elucidate the importance of this part of the genome in this virus. Doing that, and this is very interesting, we were able really, by splitting the genome into different parts, looking into the sequences, we were able to find out that we have 6 functional domains in the 3A, and these domains, each of which are linked to a very important function, which is related to virulence, to infectivity. Very important. We are not really now digging into this. We're more interested into the spike because we know that the vaccines are there, but we have to know there are other very interesting parts of the virus, like the 3A, linked to infectivity, linked to apoptosis, linked to ion channel formation, and finally virus release, which is all together indicates not only transmission, but also virulence and pathogenesis and severity of disease. So we had a total, when we compared all of the genomes, and at that time we had like almost 2,700 genomes deposited. We have aligned them, and we found a total of 51 different non-synonymous mutations leading to different amino acids. We had other mutations, but that they were not important. And what was more interesting, that we found and observed micro-clonality within the ORF3A based on those non-synonymous mutations. So when you take those mutations and you try to split the virus, the variants of the virus, you will clearly see a type of microclonality that is actually linked to those mutations. And this shows you clearly the mutations which led to a change in the amino acid. And you can see that these viruses, according to these mutations, are clustering together and apart from each other by just looking at the ORF3A. And this is extremely important. And some of these mutations, by the way, could interfere with the virulence of the pathogen. And I will be highlighting this later on. So we came up with this model of the ORF3A, as I said, looking and aligning the sequences by comparing the SARS-CoV, which was our reference, to SARS-CoV-2, to the one from bat, the pangolin, and from civets because it's linked to SARS-CoV-1. You can clearly see those domains, and you can clearly track the importance of each of these domains. And what was very clear in all the models that we have studied is that this region was characterized and still characterized by having this 3 transmembrane type of orientation. And what was very clear right from the beginning was the absence of a previously detected N-terminus putative signal peptide that we had in SARS-CoV-1, but we didn't have, or it was not there in the case of the newly introduced virus. Then what we did is that we color-coded each domain, and we linked the function when it comes to virulence, when it comes to pathogenesis, to each of these domains. And I'm going to highlight these domains as well as their functions. So if we look, for example, start with domain 2. This is domain 2. Look at the sequence. You can see that in SARS-CoV-2, in the bat and the pangolin, especially in CoV-2 and the bat, you exactly have the same sequence, nothing changed. It's a bit different from the CoV-1 and a bit different from pangolin, but this is a very important domain because this is the one that actually mediates, if you want, the inflammatory response that is linked to this virus, and which is through the activation of nuclear factor kappa B, which is involved in activating the pathway that is linked to caspase, and then eventually interleukin-1 beta. So I'm not going to go into the details of the pathway, but we know now that this domain in particular, and as you can see, it is pretty much conserved, is linked to the inflammatory response as seen in SARS-CoV-1 as well as CoV-2. If you go further and you look into the third domain, you will find that this is a real interesting domain. It is involved in its homodimerization of this important protein. And what is more important, that this is a cysteine-rich region, and one particular element, which is the C133. This is very important for homodimerization, and you can see that it's conserved in all the different models that we have studied. So this is fixed and it's not changing, which highly really illustrates the importance. And what is also important, that this is also linked to a potassium channel that is associated with this important part of the genome, and some mutations like this one can lead to the loss of the potassium channel, and if this happens, then the virus will lose its ability to cause apoptosis. And this is very important. And actually, one of the isolates that, or some of the isolates that we have detected had this mutation. You can see the microclonality, the presence of this mutation, which, as I said, could create a less virulent virus, which is not capable of inducing apoptosis. Then we had the fourth domain, which is the one that is linked to caveolin binding. This is very important. It regulates the uptake and the trafficking of the 3A protein to the plasma or the endomembranes, which again, if you interfere with this process, then it's going to be kept in the Golgi, and again, it will really interfere with apoptosis as well as with infectivity and with eventually release of the virus. So this is domain 4, and again, you can see there is a high level of similarity or homogeneity when it comes to the sequences. Domain 5, it is also very conserved. Look at domain 5, it's fully conserved, and again, it has a role in trafficking. And usually, especially the 2 amino acids at the outer amino acids, these are extremely important. Any change in those amino acids will lead to targeting the 3A to lysosomal degradation, which is again something that will interfere with the virulence and the pathogenesis of the virus. Finally, the last domain that we found out is domain 6. It's made of a diacetic motif, and this diacetic motif also is very important. It's a bit different in pangolin, in the bat, and CoV-2 as compared to the other. However, it's still functional, and disrupting this diacetic motif also was found to interfere with the ability of this virus to induce the kind of pathogenesis that we are now very aware of. And as such, any change, any mutation, and pathogenesis. So very important part of the genome of the virus, highly conserved, and the fact that it is highly conserved and we have just few changes, again, indicates its importance when it comes to the survival and the ability of this virus to cause infection and to spread further away. Now, in 2 minutes, I'm just going to highlight the fact that this virus is changing a lot. We have a number of variants. We have already detected and studied the first 11 viruses or variants that Excuse me, did you say anything? Okay, so we have already a sequence, and we were able to detect the first introduction of the virus in the country, as well as link it to travel history and community transmission. I don't have more time, but I want to really end this presentation by saying we cannot determine whether we have a new variant or not by simply relying on PCR assays. This is not right. It is wrong. You might be able to detect one mutation like the 501. You might say this is the UK variant, this is the South African. This is based solely on prediction. You need to sequence the genome. You need to find all the mutations to confirm your variant. You cannot just go out and say, I have the variant when you just did a PCR assay. Molecular epidemiology is the only way for people to really find the variants, and not just PCR, and not even the spike dropouts that some people are detecting. This is all speculative. We need to do more sequencing if we want to be faster than the virus. Otherwise, the virus will keep on beating us, and we will keep this quarantine ongoing forever. Thank you very much. I hope I was able to summarize in a very short, 10 minutes, some of the things that we are doing nowadays. Thank you.
Thank you so much, Professor Tokajian. It was very interesting. I just have one question from the participants to Professor Hajir, asking, do you advise taking the vaccine at the community level, regardless of previous infection with COVID-19?
It is advised by the CDC and by different health authorities that people take the vaccine, even, by the way, even if they were infected by the virus before. It will give them a booster to their immunity, and I believe it is worth taking the vaccine.
Thank you so much, Professor Hajir. Dr. Ahmed, do we still have time for questions from the participants, please? Dr. Saeed.
Thank you, Dr. Tokajian, for this wonderful presentation also. And I really appreciate the fact that you have talked about the importance of sequencing to identify the genetic variants, because we are hearing nonsense, let's say, on TV right now. Some people in Lebanon, I believe, they are saying that SARS-CoV-2 variants can be identified by PCR, which has never been the case. Some PCR, like hybridization probes, can identify some genetic variants. However, a real-time PCR, a TACMan, can never do this. The only way to do it is to do NGS. I believe you know it so far. Unfortunately, to my knowledge, NGS does not really exist in Lebanon. We might have some sequencers, but to my knowledge, these are not yet functioning. Can you please tell me, is it right, or what is the solution that we have in Lebanon? Should we always send our samples abroad, or no? There are some labs that they have started the NGS sequencing for the SARS-CoV-2.
Thank you for the question. No, actually, we are doing NGS in the lab. We're using the 4th generation platform, which is the nanopore sequencing. And we started this in May, actually. We brought the platform, and now we are sequencing. And whenever the Ministry of Health needs our help, they are sending us the samples, and we are doing the sequencing. The idea is that we have to develop really our, if you want, ability to do more sequencing. We are doing it, but we need more and more people, not just to rely on simple PCRs and come up and say we have new variants, it's spreading, it's causing a big issue. We need to be very careful, because people really are getting very, if you want, it's just spreading panic in the community. And I don't think this is the right message that we want to really convey. But no, we are doing NGS. We have the platform, and I think we are still the only ones in the country that we are doing sequencing, local sequencing.
Okay, and sorry for my question, but we know NGS does many false positive. In my experience, it was associated with 2.5% of false positive when doing NGS. So are you validating it?
Yeah, I agree. All of the... Yes, I agree. And sometimes even if the sample that was delivered to the lab was not of good quality, you will end up not having good quality sequencing. So there are a lot of problems there. However, because NGS produces several reads, from the coverage, you will be able to estimate whether what you are seeing is right or wrong. So if you have 1,000 reads and out of the 1,000, only 20 were a different mutation, you will be able to some level of confidence, tell that this is the mutation that you are looking for.
Okay, thank you, Dr. Tokajian.
A question for Dr. Ali Hajir. Dr. Ali, they were asking, are you with us, Dr. Ali, first?
Naam. Yes, I'm with you.
Okay. Yes. The question was being repeated several times. People who have asthma, are they safe to take the vaccine?
Yes, I saw the two questions there about people taking immune suppression and people with asthma. There is no contradiction, no contraindication for asthmatics not to take the vaccine. The only contraindication so far is people with severe allergies, such as food allergies and drug allergies, and who carry their EpiPen. These are absolute contraindication for the current vaccines. Asthmatics, actually, they should benefit from the vaccine, as it will protect them from the infection, which usually is worse in patients with asthma. How about patients on immune suppression? I suggest that in both cases, before the patient takes the vaccine, should consult their consultant, their doctor. But as this vaccine is not we are talking about Pfizer and Moderna, these are not based on live viruses. I see no contraindication in taking it in patients who are receiving immunosuppression, but they should consult with their doctor first. There are other vaccines, such as the AstraZeneca, which contains a live virus. And in this case, I would caution people not to take it if they are on severe immune suppression in very high doses of immune suppression. In either case, it's worth taking the permission from your consultant and the follow-up from the consultant before you take these vaccines.
Thank you so much, Professor Hajir. Thank you, Professor Tokajian. We will end the session and we'll move to the next session that will be moderated by Dr. Fatima Saleh.
Thank you so much. Very much.
Thank you.
I think if Dr. Ayman or others have a question, we will see. And then I think we'll move on.
Okay, shall we move on then?
Thank you for your time. It was a pleasure having you, Doctor.
Good morning. So for those who have just joined us, I want to welcome you again to our webinar. It's my pleasure to be moderating the second session, which will be focusing on the preparedness and response to the COVID-19 pandemic in Lebanon. So my first speaker for this session will be Dr. Roni Zanne. Dr. Roni is currently the Associate Pharmacy Director for Clinical Services at ABMC and the founding director of the first ASHP accredited pharmacy residency in Lebanon. He earned his BS and PharmD degrees from LAU. Dr. Zanne is a board certified pharmacotherapy specialist with added qualifications in infectious diseases. He has a diploma in principles of clinical research and a master's of public health in epidemiology and biostatistics. Dr. Zanne is currently the president of the Middle East chapter of the American Clinical College of Pharmacy. In recognition of his work, he is named Fellow of ASHP and the recipient of the Order of Pharmacists of Lebanon Distinguished Service Award in 2018. Today, Dr. Zanne will be presenting his talk on assessing knowledge, attitude, practice, and preparedness of hospital pharmacists in Lebanon towards COVID-19 pandemic. So, can we please share, Dr. Ahmed, the presentation, because unfortunately, Dr. Zanne was not able to be live today. So the presentation is with Dr. Ahmed. If you can please share it. For unforeseen circumstances, Dr. Zani had an urgent meeting, so he couldn't come to our webinar.
But we are having Dr. Fatima, we are having just a technical issue. We will be solving it. We are loading the presentation.
Okay, so if the Dr. Muhammad Faris, if he is available, so maybe we can start with him. What do you think?
I am available.
Oh, great. You saved us. Okay. Hi, Dr. Faris.
Marhaba. Hi.
Shall we start with you instead of Dr. Zani, just for the presentation to be ready? Is that okay?
Of course.
So Dr. Faris, great. Dr. Faris is a biomedical researcher with polyvalent interests that include global public health. He has published over 30 publications in international peer-reviewed journals. Dr. Faris has a master's degree in neuroscience from the Lebanese University, and he is currently in the fourth and the final year of medical school at AUB, good luck for that, while concurrently finalizing his second master's thesis in sports and exercise science and medicine from University of Glasgow, UK. Please, Dr. Faris, we are all ears.
Thank you. Just 1 second. Okay. Hi, everyone. So I'll be presenting my paper entitled COVID-19 Pandemic, an Insult Over Injury for Lebanon. So our paper was supervised and directed by Dr. Abdurrahman Al-Bizri. It was published in the Journal of Community Health later in 2020, and we will talk about the events that preceded the emergence of COVID in the country. We will discuss the emergence of the first case, the vast implications that that had on the country, whether on public health, economy, education, and psychology of the Lebanese population. We will discuss the aftermath and the situation that we are facing now, and we'll end with a conclusion. So with this, the time that predecessed COVID-19 in Lebanon, it's late 2019. It was a very difficult time for the Lebanese people. It was the gateway that led to the hardships that we are facing today. It started early October, where numerous fires broke out near the mountainous area near Beirut. Hundreds of families were displaced. Millions of dollars worth of damage occurred, and even we had one loss of life. The people were very angry at the government for the lack of preparation and inaction. Later that month, the Lebanese took to the street to revolt against the corrupt political regime. That ended in turmoil, civil unrest, and all these elements, along with the political and economic instability that revolved around the country for the past several years, led to a financial crisis that was evident by the devaluation of the Lebanese lira, the collapse of the banking system, and numerous bankruptcies and failed businesses throughout the country. To add on top of all of that, on February 21st, the first COVID case arrived to Lebanon. That was met by a major reaction by the Lebanese community. There was fear of the imminent threat that just arrived to the country. There was a sense of urgency which prompted the Ministry of Health to release a press conference to discuss how will they be able to tackle the issue. And unfortunately, details about the patient was released on the media, compromising patient confidentiality and allowing it to be politically exploited for many gains. Since then, life as we know it in Lebanon has significantly been altered. We will discuss the changes to the public health sector, the economy, the education, and the psychological well-being of the Lebanese population. So with regards to direct morbidity and mortality, as of mid-June, we had around 1,432 cases of corona. That equated to 208 per million persons. Most of the cases were mild cases. Highly urbanized areas were understandably the most affected. And we had 31 deaths in total. However, if we are going to look into the public health aspect of the situation, the healthcare system was completely drained. At the start of the COVID pandemic, the public primary healthcare centers were at the forefront of facing the virus. Later on, as the more cases emerged, other hospitals started joining the fight, and all the resources of the hospitals were being diverted towards dealing with the COVID